We begin again with a very anticipated session, Pitch Perfect, de-risking your disease to drive investment and collaboration. I think as you've heard from all the earlier sessions, this notion of being able to articulate more about your disease, your condition, your disease biology, pitch to researchers, to potential investors, it's becoming more and more important. So this is a Shark Tank style session, but we like to refer to it as our dolphin tank because we're just nicer, as Michelle said. Please welcome our panel of judges who will all introduce themselves. Walt, Kowtoniuk, Maya Chopra, Craig Lipset, Mustafa Sahin, and Yale Weiss. And our first pitch will be presented by Zali Yavaro from Cure VCP. Let's give her a round of applause.
All right. Hello, everyone. My name is Zali Yavaro. I'm the Director of Scientific Engagement and Collaborations at Cure VCP Disease. We are a patient advocacy and research organization founded in 2018 with the purpose, Thank you. With the purpose of accelerating research and supporting patients with VCP-associated multisystem proteinopathy, or sometimes called VCP disease. We aim to create resources and community to foster research and promote drug development. To drive treatments forward where there currently are none. So why invest in VCP? What's interesting about VCP? We all have our favorite proteins here. So VCP is one of the most abundant proteins in the human body. It's a very highly conserved and essential protein. It's involved in a whole bunch of different cellular processes ranging from protein aggregation, protein quality control, cell cycle autophagy, DNA replication and repair, and many more. Sometimes we think, what does VCP not do? And VCP is expressed in all tissues in the human body, and it's been implicated in a broad array of diseases in different tissues, from brain, muscle, bone, and also into cardiac, cancer, and ophthalmology. It is also implicated in an autosomal dominant inherited genetic condition that is adult onset. It's associated with single mutations in the valicin-containing protein gene, encoding for VCP. And so VCP is a multisystem disorder with a diverse disease presentation. So it manifests with muscle disease in around 90% of our patients. A little less than half have Paget's disease of bone, and around a third have frontal temporal dementia. It's also one of the familial causes of ALS and familial FTD, and patients can present with one or more of these conditions. Research suggests it could be amenable to multiple different modality approaches, ranging from small molecule that could point to VCP activity or its interactions with different cofactors, ASO silencing, or gene therapy. So due to involvement in many areas of disease and different functions in the cell, we believe that insights from VCP MSP could have broader implications and potential for use and impact beyond VCP MSP. All right, so what has VCP done in order to help facilitate preclinical resources for drug discovery? So we put together a whole bunch of different things ranging from cell lines, model systems, assays and data sets, and patient samples and biobanking. So we've created 2 patient-derived induced pluripotent stem cell lines, and these are from the 2 most common mutations seen in our patients, and these are the R155H and R159C. We've also created isogenic corrected controls from these 2 lines. So you have lines with an identical genetic background in order to do your experiments and have an ideal control. We've also differentiated them into relevant cell types, including motor neurons and myoblasts. We also have HEK lines that can help do some quicker experiments for those not wanting to work in primary lines, again with those 2 most common mutations, and looking at knockouts of VCP's cofactors as well. We also have multiple different model systems for VCP. These include mice, including conditional knockout in disease-relevant tissues like brain and bone, Drosophila fruit fly models with 9 different mutations available, and a muscle organoid in development with NIH that can measure force contraction and muscle fatigue. And this is using those patient-derived cell lines that we've created. We also have a series of assays that are both phenotypic and biochemical to look at various parts of VCP function. We've also been working on putting together multi-omics sort of data sets. These include proteomics, which has longitudinal serum samples from 15 patients in our natural history study, tested at 3 different time points. And we have a new initiative collecting genomic data as well, including whole genome and whole exome. We also have a biorepository. This now includes over 800 samples, ranging from plasma, whole blood, urine, dried blood spot card. And we collect from patients who have symptoms of VCP MSP, in addition to patients who are pre-symptomatic for the disease and unaffected controls. So we have controls collected in the same way that can be withdrawn from this biorepository. We've also collected tissues. We know of 16 brains with confirmed genetic diagnoses in banks worldwide, and over 200 muscle biopsies as well. So bringing together all of these different kinds of patient samples in order to facilitate preclinical research and discovery. For clinical trial readiness, we have been working to prepare our patient community by enrolling them in our VCP family registry. So we now have around 125 individuals on this platform. And to put this in context, we have a conservative prevalence estimate of around 2,000 individuals worldwide. And this, like many rare diseases, we think is widely underdiagnosed, especially in neurodegenerative spaces where it's not as readily to go to genetic testing to find a genetic cause for what you're experiencing. On this platform, we also have patient-reported outcome surveys that are completed yearly by those enrolled, and also the option to integrate with electronic health records. We've also hosted a patient FDA listening session, and I host quarterly office hours with our patients to promote education and trust in the community and scientific process. In order to support patient identification, we've also launched a genetic counseling and testing program. This is conducted virtually via telemedicine. We also have a stipend ordered to reimburse patients and remove barriers to diagnosis. We've also done significant work with our functional outcome measures, including running a 2-year natural history study with 46 patients. And for this natural history study, we tested outcome measures from other neuromuscular clinical trials looking for repeatability, reliability, and relevance in VCP MSP. And within this, we had sort of 2 different sub-studies as well. We wanted to see if these measures could be conducted remotely. This is especially important in our disease state, which has mobility issues where the burden of travel is really high. We found that we could conduct these remotely. We also collected a video measure and have a scorecard for this video measure as well. And we see significant changes within these populations within 1 year. And this has baselines published now. In addition, we have a broad KOL network. These include academics who are trained in basic and translational VCP science, and also clinicians who see patients. We've worked to put together a standard of care document using a working group of 50 of our clinicians, which includes all of the disease phenotypes and best practices for care. And as we mentioned in the last panel, we talked about the importance of bringing people together in one room for a conference. It really makes a difference. We've done this twice now, and it's really changed sort of how we approach VCP MSP research and change this field. We're committed to keep doing this and working on another conference. So in summary, VCP MSP is a serious disease with unmet need. And because it's an essential protein in cellular processes and has phenotypic overlap with other diseases, we believe there would be insights into other conditions. Our organization is committing resources in place in order to facilitate preclinical research, de-risk investment, and promote clinical trials. So Cure VCP disease, our research base, our patient population, they're motivated and they're ready to partner. Thank you.
Thank you. So
Thank you,
we
Dr.
have
Frost.
roughly 20 minutes where the five of us are meant to provide feedback and input. Gail, I'm going to come to you first, but I'll say a few more words here to filibuster and give you a second to think about what you want to start with. But I'll offer that, one of the ways I'd frame this for my fellow panelists is for us to think about a pitch being an exercise in trying to convince somebody to do something that maybe they would do something else. We all have finite time and resources. So with that lens, Yael, you work at a biotech company, you run a biotech company. It'd be great to understand how this pitch did and did not move you from a perspective of allocating your time and your resources to work on BCP.
Thanks, Walt. And for those who don't know me, I'm Yael Weiss, CEO of Mazi Therapeutics. We focus on developing therapies for neurodevelopmental disorders. And maybe I'll just give some background because Charlene was late and she didn't give her introduction and Nicole had to jump in. But when we started thinking about this session, we talked about the different types of personalities of patient advocacy groups. Some are just developing drugs. They're doing it. They're not waiting for an industry partner. And in the process of developing drugs, they need to pitch to potential investors or donors to get funding for the drug. So that's one type of pitch you would be putting in place. On the other side, there are the patient advocacy groups that are preparing a toolbox, just like Zoe just described, and are looking for partners to identify their disorder as the one they would develop a drug for. And that's a different type of pitch. You want the people who you want to collaborate with to understand what the disorder is, what the protein is, and what you have available for research. And in the middle, there are groups that are doing both. So my big message, the overhanging message would be, who are you pitching to? And Zolly, my question to you is, who are you pitching to? I'll let you answer in a minute. And tailor your presentation accordingly. Because if you're asking for money, who is sitting in the audience? Is it scientists that know everything about your protein but don't know much about drug development? Or is it investors who know a lot about drug development but not much about your protein or your disorder? So you have to tailor your pitch accordingly. So I think when we thought about the session, two pitches were chosen. One, like Zoli, that is talking about what's available from Cure VCP in case they want a partner. Mm-hmm. And the next should be a group that has a therapy and they're pitching their therapeutic approach. So this is the perspective of this session. And I'm sorry we didn't do it before you started. My apologies. So in this context, Walt, do you still want me to answer the question? I talked a lot. So Zoli, who are you presenting to?
I'm
Who is your
presenting
audience?
to industry and pharmaceutical potential partners.
Okay. So I think you did a great job describing what's available and Cure VCP has a lot available, right? The things that I was missing is an intro slide. This is the presentation. This is what I'm going to be talking about. This is what I'm going to be asking you. Think about it as I'm talking. I'm going to ask you to work on my disorder, and Walt will convince you to do it. This is the information I'm providing, but what questions do you have at the end of my presentation? Get ready, start thinking about it. So a summary slide that talks about in one bullet, what is BCP? What do you have? What are you asking for? And then jump into everything else. Okay. And then at the end, a summary slide saying the same thing. My board of directors tell me they forget from one meeting to another what I told them in the previous meeting. We have a presentation skills expert that says that people only listen in the first 2 minutes and the last 2 minutes. So take that into consideration when you're building your pitch. Grab the audience, they can fall asleep, grab them again at the end of the presentation. So that I would ask for that. And one thing that's specific for me as a biotech company, you have all these amazing tools, but what's available to me? Because I know I'll go to Mustafa, he has a cell line and I'll have to negotiate for a year to get the cell line from him, right? Because Boston Children's won't just give out the cell line. So what can I do to get access as a company to tools versus an academic that things are more readily available for them? And that's really important for me. Absolutely. Something to think about, maybe not even at the presentation, but as a side note or as a footnote for your slides.
Thank you.
I'll start with that.
Hi, I'm Mustafa Sahin. I'm the chair of neurology at Boston Children's, and I also direct the Translational Neuroscience Center. I'm going to skip the presentation tips, but really ask questions about the disease, if that's okay. About 25 years ago, I started working on a disease called tuberous sclerosis. And the reason I chose that disease as I was a junior starting child neurologist was because there was a connection between those genes that cause the disease and a specific enzyme, a kinase called mTOR in the cell. And as I was listening to your disease, I liked the hook of this particular gene or protein being involved in a number of diseases, but I didn't hear much about what this gene does in the cells or what this protein does in the cells and how it results in this very different variable presentations across the organ systems. And that to me could be an advantage, but could also be a disadvantage. And I think you need to turn that into an advantage in your case if you can find a link with something that you think is druggable, there's evidence for from other diseases where people have targeted a GTPase or a kinase or some other type of a protein, for instance. And the other question was about the variability of expression. There's a huge variability, and I couldn't tell from the natural history whether those things evolve into each other or they are completely separate arms and presentation of the disease, and whether there's a genotype-phenotype correlation, which would really allow you to stratify the patients and go after different indications, if you will, which would be, I think, potentially a better approach, a more targeted approach, and maybe a higher potential success rate.
Thank you. So I'm hearing two questions. One is a little bit more about the mechanism of the disease. How does mutations in BCP lead to this particular condition? And then also a little bit more on genotype-phenotype correlation. So yes, so in terms of like how do mutations in the VCP gene lead to VCP MSP, it's still a really big area of open research. So we know VCP is heavily involved in protein aggregation and protein quality control, and we have these protein aggregates that form in the cell. Our researchers are interested in understanding what is in those protein aggregates still. VCP is associated, increased activity is associated with earlier disease onset, but it's more complicated than just activity. It also has to do, this is something that came out of our most recent scientific conference, with where it's located in the cell. Is it able to have that activity? Is it able to break down these protein aggregates? And also, what is it interacting with? It has over 40 different cofactors. So we have some grants that are in place trying to address some of these issues, including spatial proteomics and transcriptomics and some of those muscle biopsies I mentioned. So that's a little bit about disease mechanism, sort of what open questions remain there. And then in terms of genotype phenotype, there's been a couple of studies, including our patient registry, we've got our natural history study, and then there's a VCP sort of international consortium in the UK and Spain. So they've been able to pool, I think, 250 different patients as well. So a little bit bigger in that cohort than what we have in our registry, was about 125. And they're not able to pull out like very strong genotype phenotype correlations. There are some mutations that are more specifically associated with FTD, but these seem like very specific mutations that are leading to those FTD sort of dementias.
On the knowing whether the pathogenic variants are activating or dominant negative or causing some untoward function in the cells versus a loss of function. I think that's an important decision point in how people think about potentially targeting different therapeutic platforms for the disease. Do you have a sense of where most of these fall into?
Yeah, I wish it was that simple. I wish it was as simple as gain of function or loss of function. So as I mentioned, we know that having increased VCP activity is associated with earlier disease onset, but I've been thinking about it as a change of function. So changing where VCP is localized in the cell or changing what cofactors it might be interacting with in order to have these impacts. So it's a great question, and I wish it was that simple, but in VCP MSP, it's not. but it has the benefit of attracting a lot of really, really bright minds in our space, which we're grateful for.
I'm Maya Chopra. I'm a clinician. I see mostly children with rare genetic disorders, and I've been doing so for 20 years, and in a range of health systems from Australia to China to France to here. I'm also part of the Translational Neuroscience Centre, where I work very closely with Mustafa and Liz, who you met this morning, as we think globally about those building blocks towards clinical trial readiness. So thank you, Zoli, for your presentation, and congratulations on the work. I heard very early that you were founded in 2018, so I think that's incredible progress in a really short span of time. The things that I think were very nicely highlighted in your presentation were the generalizability, which Mustafa touched on. What can we learn about this one rare genetic disorder that can inform a range of other disorders and maybe even have therapeutic implications for broader conditions? Clearly, you have a really nice toolkit being built, iPSCs with isogenic controls, mouse model, Drosophila model, a patient registry, and the building blocks towards a natural history study. I think as a clinician, I was tantalizingly on the edge of my seat because I wanted to hear more about the patients. I wanted to hear about what they present with, how long it takes for them to be diagnosed. This is an adult onset condition. I can only imagine the diagnostic odyssey and the valuable time that's being lost for these adults as they reach a diagnosis. And then I wanted to hear a really compelling argument for reversibility, that this was a progressive disorder, that if we intervene early, we have the potential to change the course of these individuals' lives. And as a pediatrician, knowing this is an autosomal dominant disorder, I wanted to know about the children who could be pre-symptomatically diagnosed, who could potentially inform biomarkers, and whether that was an active area of research. So the feedback I give you is a little bit clinical and not exactly to who you're pitching for, but I think perhaps round off the kinds of questions that we as academic collaborators would be thinking in our space. So thanks again.
Thank you.
Hi, everyone. My name is Craig Lipset. I am a recovering pharma executive. This is the part of the support group where you all say, hi, Craig, and you help me in that process. I spent a period of time at Pfizer working as the head of clinical innovation, so responsible in their drug development organization. And today I split my time between a number of different organizations, including the Decentralized Trials Research Alliance that I chair, which is focused on improving access to research. I work with a group called the Buffalo Initiative that some may be familiar with, which is helping patient organizations to advance their own molecules into the clinic. And I serve on the board for the Foundation for Sarcoidosis Research, where I'm also a patient. So I'm going to wear a few different hats, but I'm also going to mirror what Maya was mentioning, that this is a tremendous collection of resources. So kudos to you and the organization for following a very deliberate playbook. I think it's ambitious to put together both preclinical and clinical resources concurrently. Sometimes folks think a little more linear. We'll focus and worry about preclinical now. We'll get to clinical once we see some of these companies move assets along. But I think you've gotten a nice jump ahead of the curve here. You know, as I think about some of the reflections that we've heard so far, one perspective that that builds on I think comments from Yael is as a potential partner in industry, I'm thinking about your sustainability, right? I want to know that if you are doing natural history studies and collecting prospective data, that you are going to be around and solvent. And so one thing that I didn't hear is, as you're building up these tools, how are you thinking about the price to me in pharma to participate? Or if you're thinking, I want there to be no friction. I want you to be able to come in and engage in this indication and remove those barriers. And I think for most organizations like yours, I'm sure that's an interesting challenge to have to balance your own sustainability and recovering cost and being able to reinvest versus making sure that there's as little friction as possible for me to be able to come and engage. One build as well on the perspective from Mustafa and Maya, as I'm also because I'm kind of new to this indication, as a clinical trialist, I'm thinking about the endpoints. And so I'm thinking like, are they accessible? Are they readily measurable to me? Are there endpoints that the community has kind of rallied around? Or is that an additional investment of time and effort on our or my part as a drug developer to be concerned with, especially if those endpoints are ones that are hard to get or going to be particularly expensive and maybe become additional barriers to me. One last, observation, maybe it's a bit of a question, as just an ally and a friend hearing this is, I never believe in the, if you build it, they'll come. You clearly have to do what you're doing, which is hustling and getting this story out in front of many. When you're engaging with biotech, talking about preclinical and clinical makes a ton of sense. When you start to talk to larger pharma, these are different parts of the house. And so you have a lot of work to do to make sure that people know that these resources are available. And I'm kind of curious how you're organizing yourselves internally just to make sure that the story is told and is being heard in the right places.
Thank you.
Okay, on time. We got about 3, by the time I get done, maybe 2 minutes. So if you have questions for us, think about them, because there'll be another minute there at the end that you can ask one of us a question, or all of us. Walt Kowtoniuk, I serve on the board at Global Genes, father of 3, and I work at a place called Third Rock Ventures where we do early stage biotech investing. And with that lens, appreciating it's a slightly different audience than you're going for here, Zolly. I'll offer you sort of two threads of feedback. So the first is when I think about a team person pitching, I have a very simplistic framework, which is I think about command, rigor, intensity, and focus. And you clearly have command on the subject matter, clearly very rigorous. But we heard a couple of times about not having an ask, like the lack of an ask, the lack of who we are and why we exist in the world, maybe pulls me down a little bit and thinking about the focus and the intensity here. I think for anyone pitching, never hurts to ask. You might get a no, but at least ask. And then the other is just thinking through what would ultimately convince me to spend time working on, a product, a company, whatever the thing is around VCP. The two that are important for my job, I often think I have a dual mandate, which is, our efforts should make a difference in the world. They should make a difference in patients' lives. We didn't hear much about the patients. We didn't hear much about how there's this group of people whose life could be completely different, right? Think about some of the products that we celebrate now, whether it's, in the CF community or the SMA community, just, paint a picture of what these patients' lives could be like. And then the other, you mentioned the patient numbers briefly, but, and this is not necessarily your audience, but there's no sense of what kind of investments required. And ultimately, if somebody's going to give capital seeking returns, what the upside could be. Appreciating that's not exactly your audience. So that's what I would offer you as feedback. And we have 1 minute and 50 seconds. So if you have questions for anyone here or all of us, please shoot.
Thank you so much. I have a question for all of y'all that I think is relevant for people in the audience as well, about sort of the range of asks that you've heard people come with for different pitches.
It depends who you're presenting to. If you're presenting to a platform company that has a novel AAV capsid or an RNA modulating small molecule, your ask is, I'd love for you to work on my disorder. And the reasons are, we have everything you need. All you need to do is plug your X into my system. So that's an ask from a potential partner. For Walt, if you're asking for money, what are you going to do with it? And what's your plan? Because you haven't talked at all about drug development or discovery. And that was not the aim of the pitch, I understand. But if you are speaking to an investor, you need a plan. I'm going to do X, Y, Z, and 1, 2, 3 time points. So I don't think I can give specific numbers here, but it depends on where you are. And maybe we'll hear in the next presentation, we could talk about it a little bit more.
My guess is if you were to go across this group, you're going to have a set of asks that range from, I want 20 minutes of your time and the advice you can offer, all the way to $100 million to get a drug approved. Fair?
I could comment on our behalf that probably the commonest ask that we get is, can you help us with a natural history study? A well-designed, multi-center natural history study to help validate endpoints and biomarkers. And that's a big ask. And obviously the resources for that are huge. But I would say if there was one piece of advice I would have is that there always needs to be a physician champion. And that's probably been the biggest lesson for us, that if there's no physician champion who's driving that, then, you know, I think the chances of success are much lower.
Yeah, for a disease of frequency that you mentioned and the complexity, I suspect you're going to need some academic champions, because it might not be the first thing that pharma or biotech goes after. And you're going to need the resources of pharma, because they may have compounds for protein homeostasis or something like that, that they've discarded or put aside for other reasons that you could potentially repurpose and could accelerate your clinical readiness much faster. So I think in your case, I see your organization going in both directions, and it sounds like you're kind of doing that. But at the same time, given each disease, I think each of these conditions have different strengths and weaknesses, and depending on the disease, you may focus on one side or the other, or bring together a community that you need for all the needs you have.
Thank you.
Yeah, thank you very
for the
much.
feedback on the range of different perspectives.
Hello, can everyone hear me okay? Excellent. All right, thank you so much for having me here today to talk to you a little bit about hereditary hemorrhagic telangiectasia, or HHT for short. It is the second most common inherited bleeding disorder, but it's not a problem with the blood, it's a problem with the blood vessels. So you can see in the image on the bottom left that you have a capillary bed that has a lot of surface area for oxygen and nutrient exchange. In patients with HHT, you get these arteriovenous malformations or direct artery to vein connections that can be fragile and prone to rupture and hemorrhage. This could be a big problem if it's somewhere in the brain, like 15 to 25% of people with HHT, or you get reduced oxygenation, which is a problem in the lungs. 40% of people with HHT have pulmonary AVMs. And whenever I say 40% or 15 to 25%, it doesn't give you a lot of context. HHT is one of the more common of the rare disorders, and we suspect about 80,000 people in the United States have HHT and up to 1.5 million people worldwide. It is quite variable in its manifestations, and because you have blood vessels all over your body, you can have these vascular lesions anywhere. When they're smaller, they're called telangiectasias, so the third part of the name, and the telangiectasias typically occur in the mucosal linings. So about 95% of people who have HHT have chronic epistaxis or nose bleeding. The majority of those patients are chronically anemic and reliant on hematologic support through iron infusions and blood transfusions. So it's a huge healthcare burden and a huge disease burden. And there are no FDA or any kind of approved therapies to treat this disease. And we're here to change that. And that's where I'd like your help. So I want to tell you a little bit about the history of the disease and the landscape starting in 1865, whenever this disorder was first described in the literature. So just like Charlene pointed out this morning, sometimes you can have this huge gap in knowledge. And it wasn't really until Cure HHT was founded in 1991 and started advocating and bringing awareness to this disease that you can see an uptick in publications on this graph. And I apologize for not having a label on the axis, but this is a graph taken from PubMed of all of the publications in HHT over time. And you can see that it really underscores the importance of patient advocacy organizations to bring awareness, to drive research. And here we are today as a patient advocacy group whose role has been patient support for decades, now sponsoring a phase 2-3 clinical trial for potential FDA registration that we need someone to take forward for us. So history of this adventure that we found ourselves on, there's a drug called pazopanib, trade name Votrient, and this is used for kidney cancer. And there was at the company where this drug was, a doctor who was working with this therapeutic who was also very familiar with HHT. And he thought this could be a repurposed drug for this patient population that has a great need for it. The reason he thought that is because the HHT pathway is the BMP9/10 pathway. You can have mutations in the ligand, either the co-receptors or in one of the transcription factors that serve as the quiet down signal for the angiogenic pathway with vascular endothelial growth factor. So an anti-angiogenic drug that suppresses vascular growth in tumors might work for people who have HHT, who have kind of this dysregulated growth because the quiet down signal to that pathway isn't working as the way that it should. So the company who owned Votrient at the time entered a phase 1 trial to look at the effects of pazopanib on bleeding in people who had HHT, which is very exciting until the study was terminated because they sold their asset to another company, and that company decided not to take the drug forward. And not only that, they decided they didn't want anyone to take their newly acquired asset forward. It was, a business decision on their new asset that they didn't want to risk. So CureHHT, helped with the investigators on that phase 1 trial to publish the people who were far enough along to have data, and all 7 had positive results. So that means that they had a decrease in their anemia status, and then also improvement in their bleeding. At the same time, another study was published, a retrospective look at off-label use of pazopanib. All 13 of those patients also had very positive results with limited adverse events. And we knew that we needed to do something to keep this treatment alive and viable for people who had HHT. So CureHHT rallied, raised a lot of money, and had a lot of talks with the FDA, and we wrote a lot of grants. And we decided that the only way forward was to manufacture our own version of this drug. So Votrient is a 200 milligram capsule, and we wanted a dosing structure that would allow some flexibility in case there were toxicities, because this is a drug that people would have to take throughout the rest of their life. So we manufactured a low dose form of pazopanib at 25 milligrams. We facilitated pharmacokinetic and pharmacodynamic testing of that newly manufactured compound. We applied for an investigational new drug. We received an orphan drug designation and then a breakthrough therapy designation. And finally, we received federal funding from both the FDA and the DoD. So we were thrilled, right? There's no stopping us. We're actually doing this. This is crazy. And then COVID. So like a lot of research, an uphill battle pretty much every step of the way. So additional requests by regulators, there were delays in launching our sites, budget overages on every single aspect of this study, manufacturing, personnel, and then learning how to clinical trial as a patient advocacy group is really hard. It was a huge learning curve, and very persistent staff and leader, and sometimes a little stubborn, but finally, we can say as of this spring, we are fully enrolled on our phase 2/3 clinical trial for testing pazopanib to see if it would work for outcomes like bleeding and anemia in patients who have HHT. And we're enrolled across 11 sites across the country. It's a decentralized study, and we had 92 patients, 73 ended up randomized, and it is a randomized placebo-controlled clinical trial with an open label extension. We've been in talks with the FDA the entire way, so hopefully if we have a positive readout next year, we can start the NDA process. But we're a patient advocacy organization. We need a partner to take this drug forward. So that's why we're here pitching to you all today to give you a little bit of background and context. But for those who are considering this as a next step for your organization, there are some side effects, okay? So there was a lot more awareness on HHT because of this. It kind of opened the doors, so to speak, which was amazing. We had the first ever NIH funded clinical trial that had a positive result, the only clinical trial in HHT that's had a positive result using pomalidomide to understand effects on nose bleeding. And then the first ever novel drug was developed for HHT, just finished its phase 2 study, and we're hoping that they launch a global phase 3. And then we have several companies that are wanting to develop and begin studies in the next year. So HHT is, I don't know, I think it's, there's a handful of genes that are mutated in the disease, but not everyone responds to therapies in the same way. It's really going to take a toolbox of therapies. And we're still working to figure out who would be most responsive to which types of therapeutics. But I think it's safe to say that it's going to take, and because we have a large population of patients, there's plenty of room for people in a lot of different therapeutic options. But because of all of this, we created the therapeutic arm of Cure HHT, and that's what got me my job, which is great. But because we needed to support all of this growing interest, we had to grow too. And so everything that we did has been a little bit backwards from what was just presented with VCP. So this is actually what led us to develop our research assets in order to help support all of this growing interest in our disease. So we already had a good and robust patient database where they were familiar with research, they were excited about research opportunities, but now we work on marketing for studies, recruitment for studies, and we actually serve as a site for some of the non-clinical studies. And then we have a patient-reported outcomes registry, and then also we have a natural history study that's clinical in nature. We opened a biobank, and we're also developing a research app in an effort to standardize some of the patient-reported outcome collection and ease of the user experience in tracking their daily nosebleeds, because we've noticed that these companies that are interested in HHT in the space, they all want to go out and kind of basically reinvent the wheel in a slightly different way, but that doesn't really strengthen the field and the data that needs to go in front of regulators. So the app is our current project that we're working on. So all this to say, it's been a lot of work, but we took the risk so you could take the opportunity. Are you in? Thank you.
Thank you. That was excellent. We'll do the same approach. Al, you want to kick us off?
Amazing story. Thank you. And wow, you've done so much. So I'm assuming you're pitching to biotech and pharmaceutical companies that have the ability to take this program forward once the data is available.
Yes, that would be ideal. But in a partnership, if something happens, then we would want to have our asset back so that we can move it forward or find another
And that's
way.
a really important point.
Yes.
So it should be part of the pitch as well. But I have several questions that as a potential partner, I'd like to have answered in the deck. There's all timeline. When are you going to read out the trial? When will you have the data? Because if we have to start getting ready to commercialize it in 3 months, then that's not a lot of time. How much time do we have to prep for a launch?
It'll be next fall when we have a readout.
Okay. You mentioned a partner that has the compound for another use. Where are they in this whole story? Did you go back to them? What did they say? Do you have any novel intellectual property that maybe you own because of your dosage form or because of a new manufacturing process that makes you stand out versus them? Because everyone will ask about them.
Yes, exactly. And they are actually off of patent as of last year, but we have a method of use patent specifically for using pazopanib in the treatment of HHT. And we have in that method of use a dosing scheme so that, it gives that flexibility of dosing. We couldn't do a normal patent because it had already been public domain
Right.
in the literature. with the use of pazopanib in HHT.
So you have answers to all of that. That should be in your deck as well, because the first thing that people will ask, well, is what about the other company and what about intellectual property?
And then you
then-
also have the orphan drug
Yeah.
designation, which gives a little bit of that market exclusivity.
Yes, that's true. But you have to list all the regulatory strengths, and that's part of it. How many patients are there? Who are they seen by? Which clinicians? Do I already have a sales force that sees these clinicians, or do I need to build a new sales force for your drug? You need to provide me with that information. And then one last thing that I'd love to see added is a testimonial from a patient. I'm on this drug. It helped me. It changed my life. If you join us, you'll change many patients' lives as well.
Yeah, those are really excellent points. I think primarily hematologists would be the ones to administer and manage patients on this type of therapeutic. It also has some competitors, other anti-angiogenics, but they're all infusion based. So this really gives a lot of flexibility to patients and quality of life back, to not have to be tied to an infusion center.
One thing that we also like to see, and Walt can probably talk about as well, is you mentioned other people that are working on the indication. Will they have a potentially better drug? Is it worth investing in this drug if something better is coming around the corner? So we call it competitive landscape, but you should probably have a slide on that as well.
That's something that I think we would like help with developing too.
We're here for.
Yeah.
I have a soft spot for vascular anomalies. I think they're really interesting biology, and there'll be a lot of potential targets that can affect a huge number of patients. The fact that you have one of the more common vascular anomalies, I think, is a huge advantage, and you've done so much in terms of pushing the field forward. It seems to me a critical component will be showing if this first trial actually has a signal. And I didn't hear much about what outcomes you were measuring, how quantitative they were, how important they were related to the top priorities of the patients. You can imagine going after brain vascular malformations or something in the nosebleeds or something like that that's functional. So I think having a path which has been proven in a previous clinical trial gives so much more confidence in people getting involved in an area. That has really changed the field of tuberous sclerosis, for instance, or SMA is another example where if your first drug shows efficacy or at least a signal, then many other folks will be interested in that area. So giving some confidence about your natural history and thus the outcome measures you've chosen, or if there are any quantitative biomarkers that accelerate these trials, I think that would enhance the pitch.
Yes, that's a good point. Typically, we use hemoglobin as the biomarker to, kind of understand if the drug is working to raise those values. We also look at duration, intensity, and frequency of nose bleeding. GI bleeding is a little bit too difficult to measure for some of those outcomes, but we can use surrogates like iron infusion or red blood cell transfusion amounts throughout the trial and use those as endpoints as well.
Super, Cassie. Another really great presentation. Thank you so much for sharing the amazing work that your group has done. It's really impressive. Just to add a couple of points, I guess, I particularly appreciated how you highlighted this, the economic argument as a healthcare burden with admissions and transfusions. And I think that's particularly compelling, particularly in the rare disease space, as you know, where we really need to capture the attention of health economists. And again, just highlighting that you've taken an approved treatment and it's biologically relevant and how important it is to understand the biological mechanisms behind disorders to be able to pick these kinds of opportunities. Two questions. One is if you have, just to continue on about endpoints, did you have any quality of life measures as any of your endpoints?
Yes, we do have some quality of life measures as our secondary and exploratory endpoints for this study. The study that I mentioned on pomalidomide that was funded by the NIH had quality of life as one of its two primary endpoints, along with hemoglobin. So there are a couple of different HHT-specific quality of life surveys that are being used in studies, but they were not developed at the time of the design of this study.
Yeah, it just shows how you continue to learn along the way and then iterate.
Yeah.
And my next question, I guess, is on behalf of others in the audience. You know, how do you take that giant leap of faith to invest so much time and energy into one therapeutic and take a step back from diversifying and saying, this is what we're going to go for? And how did you come to that? And did that feel like a leap of faith, or was it a very natural choice?
Yeah, I mean, this was the only therapeutic really at the time, but also, you know, the feedback from the patients who had received this off-label was really all the motivation that you could need. We have a couple of stories specifically about people on this therapeutic on our website, you know, people who were literally dying, getting blood transfusions every 2 weeks, every month. And this one gentleman started pazopanib and he hasn't had a transfusion in 8 years. That's life-changing, you know, and it may have some side effects, makes your hair gray, can have some blood pressure side effects and some liver enzyme elevations, but much of that can be controlled by a dose reduction or by, you know, good blood pressure management. And I think that our patients take that compared to the alternative very readily.
Okay, thank you. And the other treatments that you mentioned in that last slide that are on the horizon, are they also anti-angiogenic pathway agents, or are they new?
Some of them
therapeutics?
are. So a couple of them are like AKT inhibitors. So the VAD44 is an AKT inhibitor. So pazopanib is a tyrosine kinase inhibitor. And then you have bevacizumab, which is Avastin. So that's a VEGF inhibitor. We have a couple of companies that are developing some agonist antibody therapies to try to restore the signaling in the pathway, but none of those have reached trial yet.
Thanks again.
Yeah.
So a first note would be, great job of dropping in words that matter to the judges, because you threw in the word that was a decentralized trial you ran. I'm gonna say that was for me. So thanks. I appreciate that. Heard. And for folks that aren't familiar, that basically means the study was designed to be more accessible, and it put a little more work on this group as the sponsor for the study. But it's kind of like burden is like another substance that can be neither created nor destroyed. It just gets shifted around. And so in this case, the burden got shifted from the participants to you.
Yeah, it's not easy for sure. And what this means by like having a decentralized trial, I guess the one thing that COVID gave us was that all of our clinical sites implemented telemedicine. So that was one less vendor that we had to contract when we started our trial. But that means that, you know, they only have 2 in-person visits, but they're getting their labs drawn every 3 weeks. So it is to make it more accessible, but you either have to have another vendor that you're contracting and paying to do at-home phlebotomy for labs, or and you have to have a central laboratory, you have to have a central pharmacy that distributes the drug to the patient's homes, or they have to go to a local Quest lab. If they don't live near a Quest or a LabCorp or something, you have to contract a small, you know, local lab. It is certainly a burden on the sponsor side, but I think that it's worth it, especially when you're talking about rare disease populations that are scattered around, right? And it's not always accessible to go to the doctor regularly like that.
I really appreciated the regulatory updates that you had in the deck. I would be curious if all of the engagement has been in the US, or if you're also looking ex-US. And just given the volatility with today's FDA, if these conversations are current as compared with folks that may not be there anymore.
Yeah, it is, you know, constant communications. We're lucky that our program officers and the people that we've really communicated with about this program are still there. So knock on wood. But we also have a patent in Europe. So our patent is in the US and Europe. And then our auditors are all going off of European specs for regulatory requirements for drug auditing and manufacturing auditing. So that way it gives a little bit more rigor, I guess, to the process, should the company that takes this on want to launch in Europe as well.
One thing, if you were coming to me as a biotech company doing this work, I'd expect to see a little bit more about the team that's involved so that I could have confidence or gee, wow, that you've got some really good technical expertise helping you, whether it's around GMP and manufacturing, whether it's around your clinical work. Clearly you have some good people. So I wouldn't be shy about including that in the deck, whether they're internal, whether they are advisors, whether they're from CROs or others that you've pulled into your house or under your umbrella. Don't be shy about calling them out, because you're borrowing expertise, and that matters to me. I guess one last question for you would be, what happens if pharma doesn't come? How far are you willing to go?
I don't know. I mean, I believe in this therapeutic. I mean, I believe in the stories that patients have that, are alive because of it. Even if this trial doesn't have a positive readout, it has at least opened the doors so that there is more on the horizon, right? Like, it's kind of like what Sonia talked about this morning. Sometimes you've got to know when to let things go, right? I think that this story has been really great, that it has brought us so much opportunity. I don't think I'm hopeful, but
Well,
don't know.
without an approval at your dose, could you still encourage off-label use with an
Yes,
existing therapeutic?
absolutely. Yes, absolutely.
That-
So we can
that's
include this
evidence-based. Yeah.
in clinical guidelines. We just might have to revert back to the Votrient form, or,
But I do agree. I'd love to see that sense of a landscape of whether it's what other molecules, what other targets. And I think you caveated it well, that the hypothesis today is that different patients will respond differently, that there is a certain amount of heterogeneity in terms of response. And so there may be room for multiple different approaches. And over time, then the ability to build up data to understand which patients and predict which patients may be right for which option.
thank you.
Thanks.
Great presentation. Same thing. I'll talk for, I don't know, roughly a minute, and then you can ask us some questions. I'll go back to the same two frameworks, looking at the presentation, the presenter presumably is an extension of your organization, obviously have command, super intense, the focus, the rigor. I'd like to see a specific ask though. Like, how much money do you need? Or what kind of, like, specifically what kind of people and time, but like, what really are you looking for? Then, like, we need help making this happen.
Yeah.
And then, back to just thinking about my day job and, that dual mandate of making a difference for patients and ultimately return for shareholders. I think the story you told in the Q&A was super compelling. Like, a patient who was transfusion dependent and is no longer, like, that should be in there. Like, the number one I think about is like, will this drug work? Well, this drug's going to work. We got some questions about for everybody in the dose. I got that, but like, we know this works. So I would definitely put that in there. Like, what a difference in that person's life, that patient's life. And then just back to the money, like, how much is it going to cost? You kind of mentioned how many, I think you mentioned there are 80,000 patients. So, there's clearly a sizable market here, but you didn't go all the way to like, what would really compel an investor to start spending time on this project.
Right. Okay. So my question that I have to you and comment to that is, how far out do you like to see that scoped, and like how because you can sort of predict out of those who require hematologic support, who might meet the criteria that we have in the trial for eligibility right now, and then project that out to population in each country. Is that what we should lead with, right? Of this is the demand, this is the opportunity in that format?
I don't have a good answer. I would say that what I would want to see is your perspective.
Okay.
Right. Because I look at, I would approach a conversation like this as purely a truth-seeking exercise in which you're providing your view of the world and your current best guess.
Okay.
And from there, it's just going to be a bit of an iterative process. It'll be a very empirical exercise.
Okay.
But
And
Walt,
another
would
thing
you
you could
expect her
do-
to see in her deck, Walt, that we think a price for this could look like X, that the margin on it would look like Y, that there's this size of a population, and so this type of return on the molecule, and you as a manufacturing partner are going to put in X dollars. How granular would you expect to see her first pass at this?
Yeah, I mean, I think that's good. I mean, I'd probably in a 10-minute pitch, one slide, and then, you know, in subsequent follow-up, there's probably an Excel sheet somewhere.
Well, I think in this specific case, it's a good question because the original drug is generic already, right? So there will be a pricing question.
Well, I'm
But-
hopeful that this 25 milligram dose is actually, you know, coupled with a PD marker, which is in the IP, and all of a sudden the 200 milligram dose doesn't work. And now you have, yeah, you know where I'm going.
Yeah, it's a good
Yeah.
point. Yeah. But one thing I would say that it's very rare that a patient advocacy group takes something so far, and to a partner, this brings a lot of value. You're there to support them. The questions that you ask, how many patients in each country, you can provide these answers. So if you don't have them now as a PAG, you can stand by your partner and help them with everything that they need that they do not have at the moment.
Yes, and that's completely our goal, is to be the best research partner to remove barriers and to just help accelerate the process for academic researchers or biotech pharma. So I think, you know, like all the research assets are a huge part of that, because it's really hard to find those samples and data whenever it's siloed in individual institutions, you know, across who knows where. But not every patient goes and sees the doctor at a research center, right? You have the majority of your patients probably seeing a local doctor who knows where.
Important that you know who's going to be in the meeting. So you reached out to a company and you said, hey, we have this opportunity. Sometimes it will be the business development team, and they need a very specific level of presentation. And sometimes they'll bring the commercial person and the clinician and the regulatory person, and then you need a very different type of presentation. So for everyone, whoever you schedule a meeting with, ask who is going to attend the meeting and tailor the presentation to the people that you know will be there. Because if they are interested, the next step will be signing a CDA and going into data room, and they'll dig much deeper. But you want to make sure that the people that are in the first meeting want to have another meeting.
That's really good advice. Would you ever recommend bringing a patient?
Oh, yeah,
Yeah.
definitely. Yeah.
Thank you.
Thank you.
That was amazing. Thank you to our presenters. That it really, I think, demonstrates how strong patient advocates can be in terms of leading. So we wanted to insert something into the program because there is late-breaking FDA guidance that was just issued today on rare disease gene therapies. And so I wanted to ask Yael to give us a quick summary of this new news.
So I have a summary of what was published today. There was an announcement that was, I think, made during the Duke RISE meeting on rare disease controls, and that there's a link to an FDA document, and the document states the following: it's a trial by the FDA to harmonize the biologics division with the drug division. So the division that is responsible for antibodies and gene therapies to work better with the division that works on ASOs and small molecules, and harmonize their approach to rare diseases. And the eligibility will be an investigative therapy that is specific to the correction of a genetic defect in question. And the drug is for a very small, rare population, less than 1,000 patients in the US, which is the first time they've ever quoted such a low number. The drug is intended to treat a known inborn genetic defect that is the major driver of the pathophysiology. So it has to be, the mechanism of action has to be known and related to the genetic mutation. The clinical course of the disease is progressive deterioration in function and leading to rapid and or significant disability or death in a relatively short time. So it is focused now on degenerative disorders. There are no other adequate alternative therapies that alter the course of the disease. So these are the criteria. Confirmatory evidence, so what you need to show to the FDA in order for you to enter this new path, and I'll say what it is in a minute, is evidence of the drug's treatment effect on the direct pathophysiology of the disease, evidence from a relevant non-clinical model, which is kind of contradicts what the agency has been trying to do to eliminate animal work, but I guess they're back to it for this, which is good. Therapeutically relevant clinical pharmacodynamic data and other clinical data, including case reports and expanded access data. And if you have all of this and you meet this criteria, the agency will consider getting you into a trial, a single arm trial, no placebo, and a single trial. So you may be able to conduct one trial with a single arm and get approval if you meet all these criteria. So this is really good news. The FDA is listening to everyone here in this room, and I hope they will expand beyond these criteria, but it's a really good start. So we'll keep following this.